Hey everybody, here is our video class lecture for section 17.3 in our book. It's on the process of speciation. But before I dig into 17.3, I just want to remind you about the Hardy Weinberg Principle because it's one of the more challenging concepts for sure in this unit. So I think the more you hear about it, the greater likelihood that it sticks. So just a reminder that the Hardy Weinberg Principle says that the allele frequency in any population should remain constant. So, in other words, the allele for brown hair in any population should remain constant if no evolution is occurring in that population. In order for that to happen, there are conditions that must be met. So, for example, there would have to be random mating. So, sexual selection that we talked about earlier, that can't be happening in order for us to meet the Hardy Weinberg principle. There needs to be a large population size. Really small populations might exhibit genetic drift, bottleneck effect, founder effect, that kind of thing. So that wouldn't work. There can't be any immigration or emigration. There can't be any mutations happening. And of course, natural selection can't be taking place. So therefore, we can probably assume that the environment is quite stable. Those are the conditions that must be met for the Hardy Weinberg principle to be at play, and therefore no evolution would be occurring. Hopefully, what you're thinking to yourself is, what are the odds of all of those things happening at the same time? And the answer is, it's quite rare, actually. So these conditions are almost never met, and therefore, many populations, maybe even most populations, are actually undergoing evolution. Now, the rate of that evolution is highly variable, right? It could be super slow, which is sometimes called gradualism, or it could happen in a really short burst, which is sometimes called punctuated equilibrium. So, another big thing about the Hardy Weinberg that sometimes students lose sight of is, why do we care? Like, why do we want to be able to do the Hardy Weinberg equation on a population? And the reason why is because we can use the Hardy Weinberg equation to compare allele frequency from one generation of a population to the next generation or 5 generations later of a population to determine if evolution is happening within that population. So, for example, how this would work. If we use the Hardy Weinberg equation on a population and determine that a particular allele, let's say the dominant allele, has a frequency of 60%, that would be p equals 0.60, then if no evolution is occurring, that is to say, if the Hardy Weinberg principle is at play, then if we run the equation on a subsequent generation of the same population, we would expect to get the exact same frequency of that allele, 60%. If we don't, if we run the Hardy Weinberg equation on a subsequent generation and the dominant allele frequency ends up at 50% or p equals 0.5 or any number other than 60%, then that means evolution is taking place. And that therefore the Hardy Weinberg equilibrium principle is not in play in that population, but rather something is going on. One of those conditions, one of these things is not happening, and therefore evolution is happening. So that's why we use, or how we can use, Hardy Weinberg. So as you focus on learning how to do the Hardy Weinberg equation, don't forget to understand why and how we use it. And there are, of course, tutorials on our Google Doc for the evolution unit that you can click on and get lots of really good information about Hardy Weinberg. So let me just give you a really quick example. Here is a data chart on a population of squirrels, and you can see they've been collecting this data for a lot of years, and they know the number of gray squirrels, and they know the number of white squirrels, and they know the total number of squirrels. And you can see these numbers change from year to year. But one of the questions we want to know in order to determine if evolution was happening is, what's the frequency of the alleles? And of course, white in this case is recessive, which means all of these white squirrels would be homozygous recessive. We know they'd be little a, little a. So that gives us information that we could use to put the Hardy Weinberg equation to use. And we could do it for each generation and see what happens to the frequency of the alleles, or we could do it, maybe for 1977 and then hop forward to 1985 and see if there's any changes. And that would give us information about what's happening to the frequency of alleles in this particular population of squirrels, which, by the way, might look completely different in another population of squirrels somewhere else, right? All right, so that's just my little quick review of Hardy Weinberg. Now, let's talk about speciation, the process of speciation, which is what this section is all about. So the big idea of speciation is that you might start with a single population of organisms, but over time, some type of reproductive isolation occurs, and we'll talk about how that reproductive isolation might occur in a moment. But as a result of that isolation, we end up with two isolated gene pools. And those gene pools may no longer look like the original gene pool. So, think about genetic drift here, right? Bottleneck events, the lack of gene flow, things like that can result in different frequencies of alleles and different alleles in general showing up in these isolated populations. And so over time, we may end up with two different species that are not capable of interbreeding with one another anymore. So that's like the big picture of speciation. So, speciation, of course, is the formation of a new species. All evolution doesn't result in the formation of a new species. Sometimes evolution is just about the change in frequency of an allele within a population, but it's still the same species. That would be microevolution. When we actually have the formation of a new species as a result of significant shifts in frequency of alleles, that's when we end up with macroevolution or speciation. So reproductive isolation, that situation when two populations can no longer interbreed, can be a result of three big categories of isolation. One is behavioral isolation, and your book gives the example of meadowlarks. So there are eastern and western meadowlarks. They are very similar birds, and in fact, their habitats overlap. But the members of these two species, they won't interbreed with one another because they use different songs to attract mates, and they don't respond to the songs of the other species. So songs are a behavior. Any kind of courtship ritual would be a behavior, and those behaviors can often result in different species not interacting with one another. So that's one way that species can become or remain reproductively isolated. Geographic isolation is another. In your book, they give the example of these two populations, the Aber and the kaibab squirrel in the Grand Canyon. So the kaibab squirrel is isolated on the north rim, separated from the rest of the Aberts, or from the Abert squirrel population, by both the Grand Canyon and the Colorado River, neither of which these squirrels are capable of crossing. So as a result, they are physically separated, which prevents interbreeding. And so they have become isolated, and now they look quite different if you look at their pictures on page 495 in your book. And they actually also exhibit some different behaviors as well. So even if they were to come back together in the same region, they may still not interbreed as a result of behavioral issues, but we're not aware of that because they are geographically isolated. And sometimes geographic isolation happens, spontaneously and quickly. So you have a single population of squirrels. All of a sudden, that river came through, separating a larger population into two smaller populations. And then each generation, due to the lack of gene flow and potentially differences in environmental conditions, evolves differently on either side of the river in this case. And then there's temporal isolation. Your book gives the example of orchids in a rainforest, that there are several very similar species of orchids, and that maybe they are physiologically capable of interbreeding or cross-pollinating, but they've solved that problem by blooming on different days, and so the different species don't bloom on the same day, and their blooms only last for a day. So this prevents cross-pollination between the different species, and that means there's no gene flow, so therefore they're able to remain separate species. All right, so that covers a good chunk of information. And then your book starts to dig into Darwin's finches in the Galapagos Islands specifically. So let's just talk about Peter and Rosemary Grant and the Galapagos finches for a minute. There is quite a number of different species of finch across the Galapagos Islands, and we'll talk about how that might have happened in a second. But right here in this diagram, this is just a sampling. There's tree finches, and within the tree finches, they have unique behaviors and anatomy. So this vegetarian finch strips bark from woody plants. Sounds like a terrible diet to me, but they are well designed to do it because they have a beak that acts like pliers. Versus this other tree finch that actually eats insects that are exposed, and it has this very like kind of needle nose, forceps-like beak that allows them to grasp very small objects or bugs. And then if you look at the ground finch, they have really different shaped beaks. Some of them feed on fruit and insects, and they have like kind of curved beak, which helps them with probing and grasping. And then there's this other one that feeds on big seeds that are hard to break, and so they have this very thick beak that's strong and very sharp so that it can kind of cut through and apply pressure. So a lot of variety among these finches, which is super interesting because we'll get to it in a second, but they all come from the same ancestor. So lots of different adaptations in beak shape and size as a result of natural selection predominantly. Now, Peter and Rosemary Grant are at Duke University. They're professor emeritus now. I guess it would be emeriti because it's plural. And they're actually still working. They still go to the Galapagos, but they did all of their work on Daphne major. They've been going for decades now, and there are so few finches on Daphne major that they literally every year that they would go for a couple of months, they would capture in mist nets basically every single finch on the island and band it. And then take measurements. I mean, they literally knew every bird on the island because the numbers weren't that big. And then one year there was a terrible drought. And they were able to, because they had been collecting all of this data for so many years, before the drought, during the drought, and after the drought, they were able to really see evolution or adaptation, a shift in the frequency of alleles happening in real time. This was amazing data for evolutionary theory. And one of the things you can see in this little graph here is the survival rate of finches after the drought based on beak size. So you can see the smallest beaks had essentially no survival. And then some of these, maybe we could call them medium sizes, they did okay. It wasn't great. It was tough, but a few of them survived. And then you could see there was a few higher percentage of these larger beaks, and then the very largest beaks had the highest survival rates. Why is that? Because it turned out most of the finches initially had very small beaks, and they ate these tiny seeds. And even though through natural mutation and variation, some of these finches ended up with larger beaks, behaviorally, they fed on these smaller seeds. This is where the group would go to eat, and that's where, even if you had a bigger beak, you hung out with your friends and your family and you ate these smaller seeds. But with the drought happening, they ate up all the small seeds, and the small seeds weren't being produced because there was no rain for the plants, but there was nobody eating the larger seeds. So there was lots of larger seeds around, and of course, as birds got desperate, they would try to eat those larger seeds, but the small-beaked birds couldn't do it. But the medium beaked birds, they were able to eat some of the larger seeds, and that gave them a slightly higher survival rate. And of course, the bigger the beak, the more you were able to eat some of these larger seeds and survive. But also what this means is these birds were hanging out together near seeds that were smaller, and these birds were hanging out, and these birds were hanging out near seeds that were bigger, which means they're more likely to mate with individuals they're hanging out with. And two large beaked birds that mate are more likely to have offspring with large beaks. And so after the drought, what we saw was that those larger beaked birds were, in fact, producing babies with larger beaks, and they were continuing to eat larger seeds. But eventually the rains came and small seeds reappeared. So this small group of smaller beaked birds, now they had a food source, so they survived. And they're hanging out with the small seeded plants with each other, and they're going to produce babies with smaller beaks. And all of a sudden, hopefully what you're imagining is something that looks like this. There was a diversion, right? We started off with this, but now we ends up with bigger beaks and smaller beaks. And as they're hanging out in different places, they are going to preferentially breed with those individuals. And who knows? Think back to genetics. There could have been linked genes. So maybe bigger beaked birds are also more likely to hop around on one foot just through sheer chance of a linked gene. And the smaller beaked birds are like, that is so weird. I would never date anyone that hops around on one foot. And so pretty soon there's a behavioral difference that's resulting in no interbreeding or gene flow between those populations. And voila, you now have two species where there used to be one as a result of a significant environmental pressure, which in this case was drought. Okay, so in your book, they go through kind of how speciation might have occurred on the Galapagos. So the first thing is the founders would have had to arrive. So founder effect, because just a couple of individuals made it from the mainland to the Galapagos Islands. Maybe they got blown off course or something. Now they are geographically isolated, so they're no longer mating with the mainland finches. So they are going to evolve differently than the mainland finches. First of all, they're in a new and different environment, but also their gene pool is different, right? Founder effect. They don't have all of the variation that the large mainland population would have. So then we see changes in the gene pools, and we may see speciation happen like I just described. Some birds may leave and go to other islands, and depending on what those islands are like, they may evolve differently than their original population that they flew away from. So then those species become behaviorally isolated. So even if they fly back to the original island, they're not going to interbreed. And so now you can have the two species living on the same island. And through competition and continued evolution, we end up with a lot of different varieties of finches on those islands. And your book goes into detail about each of these pieces on pages 496 and 497. So you can always touch base there a little bit more. All right, so that's 17.3. Hope this helps. Please do make sure that you read the chapters. Look at the 17.3 assessment. See if you can answer those questions. And then don't forget, at the end of the chapter, there's lots of questions to test your knowledge, including the Florida Benchmark Review, which kind of gives you like standardized test kind of questions. You've got to take the initiative on your learning here. All right, so good luck.